Hello, my dear students. Today's topic of discussion is polyploidy, mutation, and hybridization. Let's begin with polyploidy. So, as you all know, that each and every cell in our body contains a particular number of chromosomes, that is 2. So, that specific number of chromosome is a characteristic of each species and is known as genome. So when an organism contains more than two sets of chromosomes in the cells, that phenomenon is known as polyploidy. This polyploidy can be caused through cell differentiation or by the action of physical agents such as centrifugation, temperature shocks, which includes the condition of excessively high temperatures and excessively low temperatures like frost, or by the action of X-rays, and certain chemical agents are also responsible for polyploidy, which includes colchicine, veratrin, and mercury chloride. So what does these chemical agents do is they disturb the mitotic spindle of the dividing cells, and also when the cells divide they form four chromatids which join together to form the two pairs of chromosomes. So these chemical agents does not let that chromatid segregate and cause the non-segregation of that duplicated chromosomes. Hence, they convert the diploid cells into tetraploid cells, which means they convert the cells from two pairs of chromosomes to the cells which contain four pairs of chromosomes. So, this polyploidy is a phenomenon which is important because because it causes the formation of new species and also the adaptability of various habitats. As the production of the particular species is more, so we can test them in different environments and check their adaptability in various habitats. So this polyploidy can be caused due to two reasons, either due to doubling of chromosomes during the early stage of embryo formation or by the fusion of two male gametes with a single egg cells forming a triploid. Our next topic is mutation. A sudden heritable change in genetic material of an organism is known as mutation. So what is a heritable change? Heritable change means a change which can be transferred from parent to the offsprings. So when this kind of change occur in the genetic material of an organism, that process is known as mutation. And the individuals that show these changes are known as mutants. Also, mutagens are the factors that cause the certain mutations in the organisms. An individual that shows an altered phenotype due to mutation is known as variant. Now, what is a phenotype? Phenotype is a particular set of characters which occurs uh, by the connection of genotype with the environmental conditions. So if there is any change in that phenotype due to mutation, that particular individual showing that change is known as variant. So what are the various characteristics of mutation? As it is a general myth that mutations are harmful, but some of them can also be beneficial. For example, in the case of genetically modified plants, when the mutations occur, they cause them benefits. So, next characteristic is that they can occur in any cell or tissue of the organism. It is not restricted to a particular tissue or cell. Also, the genes that show higher rate of mutation are known as mutable genes, and these highly mutable sites with a gene are known as hotspots. Now, there are two types of mutations based upon the causes. First one is spontaneous mutation and the second one is induced or artificial mutation. Spontaneous mutation occurs naturally without any cause, without any pre-information, that is on the spot. So the rate of spontaneous mutation is generally very slow, but in the case of prokaryotes, it is as comparatively lower than the eukaryotes. So the spontaneous mutation can be caused by the action of UV rays present in the sunlight, which are seen to be causing mutation in certain bacteria. Next is induced or artificial mutation. These are the mutations produced due to treatment with either a physical or a chemical agent. So in this we use the induced mutagens for crop improvement. This process is known as mutation breeding. The example of induced or artificial mutation is the action of X-rays or alpha rays which cause mutation in particular plants. Now the second classification of mutations are based on the changes in genetic material. What changes they do cause in the genetic material helps them classify this. The first one is chromosomal mutation, which leads to the change in the amount of genetic material or its position. Second one is point mutation. This is the mutation that occurs within a gene of a DNA molecule. This can be inheritable and can become permanent. That is, it is restricted to a particular parent organism or an offspring and does not get inherited again. So, what are the types of mutagens that cause these mutations? They are classified into two. First one is physical mutagens and second one is chemical mutagens. Physical mutagens are present in the form of radiations that can be ionizing and non-ionizing. The examples of ionizing radiations or mutagens is alpha rays or X-rays, while of non-ionizing is UV rays. Then coming to the chemical mutagens, these are certain chemical agents that cause mutations in our body, for example formaldehyde or alkylating agents. Then what is the significance of mutation? Why is it necessary to study them, or why it is necessary to occur them? Okay. They cause the resistance of a plant towards a disease, so they are necessary for us. Also they help in development of new varieties and can also cause morphological and anatomical changes in the plant, leading to improving their quality. So they are necessary. Next is hybridization. The process of formation of hybrids is known as hybridization. हाइब्रिडाइजेशन में हम लोग क्या करेंगे, दो स्पीशीज लेंगे या वेराइटीज लेंगे जिनका एक एक सेट ऑफ कैरेक्टर्स डिफर करता है और उन दोनों को क्रॉस कराएंगे यानी मिक्स करेंगे. किस लिए मिक्स करेंगे? ताकि हमारे पास एक सिंगल वेराइटी आ सके जिसमें वो दोनों क्वालिटीज हों जो डिफरेंट डिफरेंट थी उन कैरेक्टर्स उन सेट्स में. So the resultant hybrids can be monohybrids that have one pair of different characters, dihybrids that have two pairs of different characters, or polyhybrids, as the name indicates, more than two pairs of different characters. So hybridization helps us in inducing a single variety, the favorable characters of other varieties or species. इसमें हम लोग क्या करते हैं, एक सिंगल वेराइटी बनाते हैं जिसमें हमारे डिजायर्ड कैरेक्टरिस्टिक्स हों बाकी दोनों स्पीशीज के. और इससे हम लोग क्या करते हैं कि कई बार जो करेक्टर्स दोनों ही पेरेंट सेट्स में नहीं होते वो भी इंड्यूस हो जाते हैं. सो प्लांट ऑफ सम स्पीशीज कैन क्रॉस, सेम स्पीशीज कैन क्रॉस इजीली and produce a fertile progeny. So hybridization of plant occurs in nature through various mechanisms, but majorly by cross pollination, that is by the transfer of pollen grains, जो कि इंसेक्ट्स की हेल्प से भी हो सकता है या वेंट की हेल्प से भी हो सकता है. An example of hybridization is Vidania somnifera, जिसे हम अश्वगंधा भी कहते हैं. उसकी दो वेराइटीज हैं यहाँ पे, इजरायली chemotype 2 and South African chemotype, which have been crossed or have been hybridized, and they have formed a new variety which contains 3 new methanolites, that is the active constituent present in it. उसकी 3 नई types को उसमें induce किया गया है by hybridization. Also an important development in hybridization is nowadays is through the medium of tissue culture, that is mainly by protoplast culture, which you will study further in the next chapters. Thank you.